good morning and good afternoon in Barcelona, and good morning, early morning in USA, and good past midnight in Australia. I hope you are enjoying this exciting meeting, and let's go to the last day, and let's go for the last session on therapies. And now it's my great pleasure to introduce Kim Hensley. She's an expert neuroscientist, head of Childhood Dementia Research Group in Flinders University in South Australia. And she will present today her work modeling MPS disorders, novel insight into disease mechanism and identification of potential treatments. Now it's your turn, Professor Hensley. Thank you.
Thank you very much. Thank you. I really appreciate the opportunity to be with you today, and I'd like to thank the organisers for that opportunity. And I'd also like to acknowledge that I'm coming to you from South Australia, which is Ghana country. I would like to pay my respects to elders past, present and emerging. Now, these are some of the usual suspects when you think of MPS models that we use to understand the disease and to test treatments. And I am going to talk to you about one of these. We use mice in a lot of our work, but I'm also going to talk to you about some of the more unusual species with MPS, and in particular, flies. And here I'm talking about the vinegar fly that we are starting to use in our work. And I will also be reflecting on a little bit of material that we're starting to obtain from patient cell-derived models. Now, starting with the mice, our vision of the future for children born with Sanfilippo is this. The baby will undergo newborn screening, and if they're unaffected, there will be no further action, of course. But if there's a diagnosis of Sanfilippo syndrome, there'll be a prognostic tool administered, and this will determine whether that child is on an early onset, rapidly progressing path or a later onset, slowly progressing path. Now, at the moment, newborn screening is possible, but it is not utilised in many, if any, places around the world. So we are not diagnosing Sanfilippo at birth. We are also not in the position to use good prognostic tools. While there are some mutations that can give us an idea of the future for that child, I think we need better tools so that at birth we can work out what the future holds and when therapies need to be administered. Now, an ideal prognostic tool for Sanfilippo syndrome will accurately predict disease course in a non-invasive way. We believe it needs to be widely available and therefore cheap, so that these prognostic tools are not limited to the well-developed world. And ideally, they'll also monitor therapeutic efficacy. Now, we've heard quite a lot in the meeting so far about the lovely data that comes out of natural history studies that allows us to understand the disease process in Sanfilippo syndrome. And what we do know from MRI studies is that shown in red here, the Sanfilippo type A, and blue, the Sanfilippo type B patient brain undergoes shrinking, so atrophy. That it affects the cerebral cortex, the grey matter, the corpus callosum, and the lateral ventricles increase in size. But what we don't often talk about is retinal atrophy, and particularly photoreceptor cells that are located down here in the outer nuclear layer of the retina. And there's some evidence in the literature, mostly from post-mortem studies, that suggest that those cells die off and that the retinal function becomes affected as a result, and dark vision becomes affected in particular. Now, retinal imaging is able to be undertaken with a very commonly used technique called optical coherence tomography, or OCT. It uses reflected light to visualise the individual retinal layers, and there's a pseudo-coloured image shown down there on the right. Essentially, it's non-invasive. The patient is pretty much having a photo taken of their retina. It's also widely available, and many of you have probably had it happen when you've gone in for eye checks. And importantly, if we're considering using this technology in newborns or in neonates or young children, it needs to be applicable. And this picture here, shown on the left of your screen, number B, or letter B, is actually a baby in a humidity crib having an OCT image taken of their retina. So it is possible to do this in little ones. It's also possible to do it in young children, with a handheld device, and the images there are compared with reference images. So our thinking is that if the retina is part of the central nervous system, and if there is a relationship that we can understand between retinal and brain degeneration in Sanfilippo, then imaging of the retina with something like OCT or other imaging modalities may enable us to visualise what happens in the brain. And this is a bit of a schema that has been our sort of working model. So we'd see newborn screening followed by a diagnosis. We would then go in with something like OCT, and any changes present there, if the retinal degeneration can parallel brain degeneration, would then indicate a rapidly progressing form of the disease if it's present in the newborn. You'd then obviously go in and treat very early and monitor with OCT. If there's no change in the retina, then you might continue to monitor. So we've very recently published a study showing the relationship between retinal disease and brain disease in Sanfilippo type A mice. What you can see here in the graphs at the top is that heparan sulfate accumulation is very similar. The rate of it, the onset of it, is similar in the retina and in the brain. And we also get expansion of the endosomal lysosomal system, as you might expect, in a similar manner. We also looked at the inflammatory response, and you can see that what happens in the brain parallels what happens in the retina when we look at both the microglial response and the astroglial response. So in our view, the time of appearance of those changes and also their rate of progression is similar in brain and retina. So we predict that you could use non-invasive imaging of retina to infer brain disease. But can that imaging be used to monitor treatment? Well, this is some unpublished data that we've obtained in a collaboration with Bob Casson at the University of Adelaide and other colleagues there. And what we've done is we've the AAV9 sulfamidase vector into those mice using IV injections. The control mice were given vehicles. The mice were treated as newborns, and we gave those vectors using the temporal vein injections on both day 1 and day 5. And the reason for that is because the eye is still developing at birth, and so we wanted to hit as many cells as possible with those sequential injections. We then did some OCT imaging, euthanized various cohorts of mice, and we looked at substrate accumulation, inflammation, and thinning of the retina. And what you can see here is that the vector treatment prevents both retinal and brain disease. So accumulation of heparan sulfate compared to we're looking here at the blue and the green groups. We saw prevention of heparan sulfate accumulation in the retina and in the brain of those mice. We saw prevention of the activation of the immune response in the mouse eye as well as in the brain regions we examined. And most excitingly, we saw that that vector prevented photoreceptor death that was able to be visualised using OCT. And so here is a mouse on the left there that is being imaged. He's anaesthetised, so he can't run away. But in children, of course, we would ideally not be anaesthetising. And we saw when we looked at those animals on a repeated basis, so these are the same mice being repeated as the disease progresses, we saw a thinning of the total retina and also the outer nuclear layer where the photoreceptor cells are. And we can pick up those changes as early as 10 weeks of age, so very early in the disease course. And this, we took measurements at 2 different distances from the optic nerve head. And then when we turn to the effect of treatment, this is the OCT data. So using that retinal imaging, we were able to determine that there was a thinning of the retina in the vehicle-treated animals and that this was prevented in the animals treated with AAV sulfamidase. This was an effect observed at both 12 and 20 weeks post-treatment and at 2 different distances from the optic nerve head. So in our view, OCT can be used potentially to predict disease course and monitor the efficacy of treatments, but that's providing they access both the brain and the retina, and not all treatment types will. We have done a little bit of work in attempting to get OCTs from humans, and if you'd like to talk about that in the questions, I'd be very happy to answer that. But for now, what I'll move to is some of the other model systems, and that is Drosophila, so the vinegar fly. I think we were probably the first to describe an MPS model of in a Drosophila, and that was back in 2018. And this is a collaborative study led by Louise O'Keefe at the University of Adelaide. Since then, there have been models created that reflect MPS 7. This is work coming out of West Bengal in India. And there's also an MPS 2 fly that's fairly recently been described. And this is a number of European groups. And you're probably sitting there thinking, well, why on earth would we go to Drosophila when we have beautiful mouse and large animal models? Well, the genome is very homologous to humans. Disease-causing genes have orthologs in Drosophila. And Drosophila compared to mice, I can tell you because I get the bills, Drosophila are very cheap compared to mice. Ethically, there are fewer restrictions. They reproduce very fast. And most importantly of all, there are amazing genetic tools that are available to be used with Drosophila. And as I'll show you in a moment, they can also be used for drug screening. So this is some of the work that we've undertaken in Sanfilippo type A flies. And what we're doing here is we're putting flies into a geotaxis test. And so this involves putting them into a cylinder and asking how readily can they climb up the cylinder wall. And here we're testing wild type flies, MPS 3A flies, and then MPS 3A flies that have SOD1, an antioxidant enzyme, increased with ectopic expression. We've also looked at SOD2 flies. And what you can see in the graphs in KNL is that SOD1 flies, so this is flies that just have more SOD1 or SOD2, have no phenotype compared to control. They can both climb the same amount of distance up that measuring cylinder. The Sanfilippo flies, on the other hand, the CG14291s, they struggle to get up the cylinder, so they have less of that white bar in the graph there. They can, fewer of them can go to the top of the cylinder. And when we actually introduce more SOD1 into the system in a Sanfilippo fly, we actually make the flies less able to climb up the cylinder. And we can talk about some the reason for that if you'd like in the chat. But essentially what we're doing here is a genetic modifier screen. So we're taking Sanfilippo flies and introducing either more or less of another gene and asking what it does to the phenotype. So it's quite a powerful way of asking which genes or gene pathways affect the disease. And also we can identify potential intervention points. Now, MPS 7 flies have been used to examine intervention points. And in this case, it's a polyphenol called resveratrol. And the MPS 7 flies were found to have fewer dopaminergic neurons in their brain. And this was able to be prevented. The loss of those dopaminergic neurons was able to be prevented with resveratrol. This was shown to be due to reduction in apoptosis, and it led to improved climbing ability in that the cylinder test, the motor test in those flies. But you can actually upscale this in flies. And what people are doing, not to my knowledge in the MPS field, but this is somewhere we need to go, is in large-scale screening of drugs. So the flies can be put into multi-well plates that have been seeded with fly food that has a variety of different drugs embedded in it. The flies can be modified so that they express either a luminescence or a GFP marker, and this might be a marker that enables you to measure autophagic flux. And then the flies exposed to the different drugs could potentially have up or down-regulated autophagic flux. And it's a really nice way of identifying a large number of drugs that may modify a phenotype in flies very fast. But we haven't quite got there yet. But what we have done is create a new Drosophila model of MPS 3C. Again, this is work with Louise O'Keefe at the University of Adelaide. The work was done by Laura Hewson, who is a PhD student. What you can see in the graphs on the left there is that by knocking down HGSNAT, we get an increase in heparan sulfate, shown in B and C, in both unaged and aged flies. We see expansion, shown in D and E, in the endolysosomal system, and this leads to impact on the fly's climbing ability. So we have fewer flies with MPS 3 that are able to actually climb the length of that measuring cylinder, and this phenotype worsens with age. What we have gone on to do, and this is a poster that was presented yesterday actually, but still available to you, Newman et al., and this is work led by a postdoc in my group, Dr. Adeline Lau. Adeline has arranged studies where the flies have been exposed to an SRT drug, and you can see there the impact of that drug on heparan sulfate in the flies. So this is one way we're starting to use these flies in our work. And just to finish up, one more slide, just to let you know about a study that we're undertaking now in Adelaide. It's been going for around about a year or so. This is a personalised medicine approach for Australian children with Sanfilippo. We have recruited 5 patients and age and gender matched controls, and we have taken a skin sample from each of those patients and controls. The skin has been grown in the lab so that we have pure fibroblast cultures. From there, we have reprogrammed them to induce pluripotent stem cells. They have been differentiated into neurons, and we're now studying them in the dish. We're looking at different functional characteristics and structural characteristics, but the ultimate goal here is once we have a phenotype, those cells will be exposed to a large number of drugs, and a high throughput screen will hopefully identify drugs in those plates that can be repurposed for use in Sanfilippo type A patients. That's, you know, individual patients. We're expecting that there may well be drugs that are only potentially of interest to one patient, and other drugs that are of interest to other patients or larger groups of patients. So just in summary, because of their size and because of a lot of different similar characteristics to humans, many of us use mice and dogs and cats, rats with MPS, and certainly our work with mice seems to suggest that we may be able to use the retina to tell us about what's going on in the brain in Sanfilippo syndrome. However, those models have pros, they also have their cons. Cost and ethical issues are just some of them. And so I think you're going to see an increasing amount of work being done in fly and zebrafish models that can tell us more about the disease processes and also can identify new opportunities for therapeutic intervention. And I'm not sure if we're the first, but I do hope there is a move towards personalised medicine for patients with Sanfilippo and other childhood dementias. So with that, I thank you very much for your attention.
Thank you, Kim, for your presentation. It's very appreciated for the clinicians. What are you doing in the lab? And I think we have one question from one clinician. Do you think we should control retina in patients with MPS 3? Because I think we are not doing everywhere.
Yeah, I think it would be fascinating, but I can tell you from the little bit of experience we have, it's not going to be easy in patients with Sanfilippo syndrome. Where we're imagining this to be used is actually in a pre-symptomatic patient. The work that we have done has been tricky. It's been tricky. Patients with Sanfilippo syndrome are reluctant to sit still, and it's quite difficult for them to comply. So we would see this being used in a pre-symptomatic manner in the future.
Yeah, but it's a good thing to do OTC in patients, because we are not doing usually, and it would be a good biomarker for trials. And we have other question. Do you think, hi Kim, very beautiful work for IPC cells work. Do you plan to test your drugs in vivo? As drugs which work in vitro may not work in vivo, it is not easy to do OTC in grown-up children with MPS 3 from someone.
Yeah. No, that's a very good question. Well, we have a model of MPS 3 in the lab, so it would be possible for us to test these drugs in the mice and to ask whether they do have a phenotypic change. I think it is a really good question and a really important thing for us do before moving a drug into patients.
Mm-hmm. So there is no more questions. So thank you. Oh, sorry. Can we use any drugs of Parkinson in MPS 2 or MPS 3?
Can we, sorry, what was?
Can we use any drugs of Parkinson in MPS 2 or MPS 3?
Yeah, well, I guess that's actually what we're trying to ask. We're aiming to use a neuroactive drug panel that will have drugs in it that are used in Parkinson's disease and other brain diseases or central nervous system diseases. So that, yeah, I guess we will find out whether any of those drugs modify changes in pathways that overlap between Parkinson's and
We have to wait for your work in these 5 patients and APC cells. And I think it's all. Thank you very much for your nice talk. Now I will introduce the next speaker, Qin Yen. Qin Chen is a professor of Department of Pediatrics in Medical Center in Dallas in USA. And he's working on development of gene therapies for many neurological disorders, especially in aspartate glucosaminuria and in CLN7. He will present now the work, preclinical gene therapy with adenovirus in aspartate glucosaminuria mice, and provides evidence for clinical translation. It's your time, Professor Chen.
Okay, so can you hear me now? Okay, great. Okay, so I'm really glad to be here to present our have the chance to present our preclinical results in rodents, which strongly support clinical evaluation of AAV9 gene therapy for AGU patients. So AGU, well, AGU is an autosomal recessive lysosomal storage disorder, which is caused by the absence of a functional enzyme aspartate glucosaminidase, or AGA in brief. AGA is a lysosomal square protein, so that we can expect there's a cross extraction when we do a treatment. The lack of functional enzyme AGA results in accumulation of AGA substrate as aspartate glucosamine in various tissues and body fluids. AGU patient is characterized by the progressive neurologic disorder with mental retardation and peripheral symptoms, including skeletal and joint abnormalities. There's no specific cure currently for AGU patient. The patient care is still limited to medical, social, and educational rehabilitation. Over the last few decades, AAV9 or adeno-associated virus 9 has a very good efficacy and safety records in preclinical studies and in a few hundreds of clinical trials. In our lab, we have extensive experiences in studying AAV9 as a vector to treat neurological disorders in animal models. My PI, Dr. Stephen Gray, initiated the first in human phase 1 clinical trial using intrathecal or IT AAV9 for giant axonal neuropathy, or GAN in brief, in 2015 at the National Institutes of Health Clinical Center. Currently in the lab, we are working on 2 dozens of neurological disorders with half a dozen of the project at the stage of IND enabling, including this AGU and the CN7, which I have been working on. We hypothesize that intravenous or IV or intrathecal or IT in brief, delivery of self-complementary AAV9 AGA vector imparts therapeutic benefits to AGU mice. To test this hypothesis, we generated the IO vector, which is shown in panel A. To test this vector, we injected mice, both male and female mice, AGA knockout mice, either intravenously IV or IT at 2 months old, which is pre-symptomatic stage, or 6 months old, which is early symptomatic stage. For IV, we injected a bolus of 200 microliter of either high 1 times 10 to the E12 or low 2 times 10 to the 11 Vg per mouse. For IT, a 5 microliter of bolus of either low 2 times 10 to the 10 middle dose 1 times 10 to the 11 or high dose 1 times 10 to the 12 Vg per mouse with dose. 1 week, 1 month, 6 months, 12 months, or 18 months. At months, we collected samples from mouse to measure AGA activity, AGA substrate concentration. Behavior tests and the brain imaging were carried out at 14 months old and 16 months old respectively. At the end of the experiment, when the mice reaches 18 months old, histology, including immunohistochemistry, were performed. This figure shows that after treatment, the AGA activity in serum and tissue and the CSF. As you can see in this knockout mice, serum AGA activity is at the level of 0.1 nanomolar per hour per ml serum. Our heterozygote mice is around 1. And then our treatment dose dependently increase the serum AGA activity rapidly and sustainedly throughout the experiment. The same phenomena can be seen in tissues, including peripheral tissue, heart, liver, and the central nervous tissue, brain, cervical spinal cord, and the CSF. In knockout mice, we also see significant accumulated AGA substrate in serum, shown in pink here, as well as in CSF. Our treatment, especially high dose IV and IT, almost completely eliminate the AGA substrate accumulation. As early as 1 month, as early as 1 week post the injection. The similar can be seen in the CSF AGA substrate in clearance. Behavior test, including the open field test, showing that our knockout mice have poor performance in terms of travel distance, mobility, and immobility. Our treatment, especially high dose of IT and IV, almost completely rescue the abnormal behavior. Small animal imaging of the brain with MRI didn't detect any structural changes in various parts of the brain. Also, there's no change of the brain weight across the groups. However, histology staining, this showing that knockout mouse has significantly reduced Purkinje cells in cerebellum, our treatment can dose-dependently preserve these Purkinje cells. This is a quantification of the staining, HE staining, which also further confirmed by immunohistochemistry staining using antibody against the more specific marker calvinin. So, as consistent with the reported results, the AGA knockout mice has increased gliosis shown by GFAP staining. Our treatment also dose-dependently reduces this brain gliosis, especially at the level of high-dose IT1E11 or IV 2E10, 2E11. So this is a quantification of the staining shown in previous slides. So in general, overall summary of our AGU project. So in this AGA knockout mice, we concluded that our vector AV9 AGA vector is effective because it increases AGA's activity in various body fluids, including serum, CSF, and various tissues, including both central and peripheral. This vector also eliminates AGA substrate accumulation in serum, CSF, and I didn't show data in also in tissues. And this vector also rescue abnormal behaviors, including open field test. This treatment also preserves the Purkinje cell number and reduce gliosis. All data I showed you were mice treated at 6 months old, but we also see similar data from the mice treated at 2 months old. We also concluded from our collected data showing that AV9 AGA is well tolerated. As you probably noticed, that at as high as 1E12 IT, we didn't see any toxicity or side effects from the mice. Which, if we translate it to the human, 1E12 is almost 4E15. So far, the highest dose ever dosed in human is 1E15. So that's almost 4 times as higher, which give us a safe margin we can dose in human. So all these results put together provides strong evidence for clinical trial. We published this data last year in the Journal of Molecular Therapy. If you need more detail, you can go there and then read the paper. The IND application now is in license to the company called Neurogene in New York City. They are now preparing for IND application. With this, I'd like to thank my PI, Dr. Stephen Gray, who is a very decent mentor. My Dr. Virgin Minasan is our pediatric neurologist, is our big boss here. I also like to thank my colleagues, both previously at UNC and then currently at the UTSW. Our collaborators at UNC, UTSW, Jack's Laboratory, Greenwood Genetic Center, Virogene, Charles River Laboratory, and our funding sources, including Rare Trade Hope Fund, ATM Terrason, UNC Track, Neurogene. And I thank you for your attention. I'm ready for questions you might have. Thank you very much.
Thank you, Dr. Chin, for your presentation. It's very nice to see that in ultra-rare disease like aspartate glucosamine
Mhm.
there are investigation. I've never seen a patient with this pathology, but yesterday with Dr. Pineda we were talking, and I think in Spain there is only 3 or 4 patients in, I think, in 30 years. But
Okay.
it's nice to see that you are investigating ultra-rare disease.
Yeah,
So,
I mean, go ahead.
no, please.
Yeah, we are very lucky. So actually, Stephen Gray was reached by Julia Terravaren, who actually has 2 sons with this AGU condition, which is a little bit sad. But Steve and eventually, immediately, like I want to take the project, and then I started this project almost 5 years ago now. So it kind of, I'm happy to have the chance to work on this disease. And based on that disease, we have been working on many other neurologic diseases, which I hope eventually, actually CN7 disease, we just dosed the patient 2 months ago, the Batten disease, type 7. But we have multiple diseases now going to the clinical trial. And I hope this AGU project also soon will go to there, but we still have some work to do, including toxicity and in large animal, and also they are working on natural health study.
History study, yes.
History study. So hopefully this can go to clinical soon.
We hope soon. So we have one question. Dr. Chen, do you think your approach with adenovirus can be applied also to non-secreted enzymes, membrane-bound enzymes, like MPS 3C, for example?
Yeah, I mean, as I mentioned, the AGU is a secreted protein. Clearly, we show evidence. The other one, the second project I take is actually going faster, which is CN7, which is a lysosomal membrane-bound protein. We just dosed the 1 patient in May. So our preclinical data is very striking, really successful for the membrane-bound protein, which is beyond our expectation, but it happened. So I guess even membrane-bound protein, you can do this similar treatment. So that's good to us. So our first patient dosed in May, which is almost 2 months ago. She's very good now. Yeah, we were judged by at 3 months post-injection, and then we were dosed the second patient scheduled in August 25th. So that's very exciting for us. So I guess the membrane-bound protein is also the candidates for the treatment, I guess.
Thank you. And other question, at what age could patients enter in the trial? People are asking, and is this applicable for other disease? At what age do you think that patients could enter in the trial? Could they enter in the trial?
According to our data, generally speaking, for gene therapy or other therapies, it's this is a neurodegenerative disease. So our rule is the earlier the better. Everybody probably agree with that. But in this particular AGU, the development is kind of slow. It's not like CN7. A lot of patients dies at early childhood or less than 10 years old. But in this AGU, many patients can still survive to their 20s or even 30s. So according to our data, this suggested that even treated at the symptomatic stage, like the 6 months old I presented, there's still some rescues. So I hope we can broadly use this to patients, or at least to the teenagers or even early adults.
Wow.
Yeah, I think this would be possible, but maybe not in CN7 because the patient develops much faster. And in that study, we show that at the early symptomatic stage, the they barely had any rescue. But in AGU, we see some benefits at old age.
I don't know if there is other question, please. No, there is no question. So I think we can move to the other speaker. But thank you very much, Professor Chen, because it was very, very interesting. Thank you very much. And let's move now to the next speaker is Professor Lila Simonaro. She is professor on genetics and genomic science in Icahn School of Medicine at Mount Sinai in USA. And her group is working for many years on the pathophysiology and treatment of IMPS using animal models. And she has been working with the drug pentosan polysulfate for treatment of IMPS patients, mice, and with a trial in IMPS patients. And now she will talk about the workings and the cannabinoid system as a novel treatment of IMPS disorders and other lysosomal storage disease. Thank you very much, and it's your turn.
Okay, thank you very much for the introduction, and thank you very much for the invitation. And it's always my pleasure to present our work. And I hope next time the meeting we could all meet in person. So my lab is always looking for systems that have common pathology in the LSDs, and actually are common causes, so that we could find drugs that we could repurpose and that we could use across the board for different LSDs. It's my disclosures. So what are the new, this is not working. Okay, one of these systems that we started looking at recently is the endocannabinoid system. And this is just a brief introduction to this system and exactly what it is. So it's a ubiquitous signaling system that's composed of several lipid mediators that interact with G protein coupled receptors. There are two main lipid mediators, AEA and 2AG, and these are the main receptor, and the main receptors are CB1 and CB2. Now, CB1 is primarily expressed in neurons, while CB2 is primarily expressed in the periphery, in the microglia and macrophages. CB2 expression is upregulated in the CNS in response to stress, where, as I mentioned, is primarily found in microglial cells. So this is just a schematic of the system showing the different receptors in the different parts of the body. As I mentioned, the CB1 receptors are mostly in the CNS, but it also does appear in other systems, such as the reproductive system, the eye, and the heart and the lung. And on the other hand, the CB2 receptors are mostly in the immune system, liver, bones, spleen, colon, and pancreas. And these receptors affect many different responses in the body, anywhere from stress to inflammation, memory, mood, all the way down to appetite and many other responses. So many drugs have been developed to target CB1 and CB2 receptors, and several of them have actually entered clinical trials. Most of the trials focus on drugs that activate CB1 receptors to impact diseases such as addiction and PTSD, or to reduce neuropathic pain. Animal model studies have shown that CB2 acting drugs can reduce inflammation, promote bone healing. Therefore, they can impact pathologies that are very common to the LSDs. Now, in 2001, Sanchez and colleagues showed that activation of CB1 using THC, which is the active cannabinoid in cannabis, leads to sphingomyelin reduction. Therefore, it's been suggested, based on Sanchez's work, that activation of CB1 could be considered for a treatment for type A/B Niemann-Pick disease, which is actually due to the accumulation of sphingomyelin. However, one has to consider that CB1 agonists have a psychotropic effect, which limits their widespread use, especially in children. So investigators have developed an indirect approach using inhibitors of the enzyme fatty acid amide hydrolase, also known as FA, which leads to the elevation of the endocannabinoids that can activate CB1 in a more controlled way. So one of these drugs is this PF04557845, which I'm going to now just call it PF. The reason it's PF is because it was developed by Pfizer. And this is a prototype of a FAR inhibitor that's now been used for in trials for and studies for Niemann-Pick disease. So it's a prototypic inhibitor that it's also been used in other clinical trials for complex neurological diseases. Importantly, it crosses the blood-brain barrier, and it's been shown to be very safe in humans. It's actually now been studied in the mouse model of type A and B Niemann-Pick by Dr. Maria Letzmer, actually in Madrid, together with Dr. Schuckman and colleagues. They found that treating these mice with this FA inhibitor led to a reduction of sphingomyelin in the neurons and brain. It improved behavioral tests such as Rotarod and the Y maze. It delayed weight loss. It significantly extended the lifespan of these mice. And in addition, they found that in mice of type C Niemann-Pick, which is a very similar disease, but it's involved in cholesterol transport, they found a reduction not only in sphingomyelin, but also in cholesterol. So I'm just going to briefly review the results from the Bartol paper, which was from these studies with Dr. Letzmer and Schuckman. This is showing sphingomyelin reduction in the neurons of these type AB Niemann-Pick mice using the different types of these FA inhibitors. The one I just spoke about, the PF, showed the most reduction in sphingomyelin, but also two other FA inhibitors that were used also showed a reduction in sphingomyelin in the neurons of these mice. In addition, as I mentioned, they looked at behavioral studies. If one just looks at the inverted triangles, these blue triangles, these were the knockout mice that were treated with the FA inhibitor. And one could see that there was an improvement in the Rotarod ability of these mice. They also looked at the Y maze. And again, if one just looks at the white column here, this is the knockout mouse that received the FA inhibitor. One can see that there is a significant improvement in the Y maze with these mice with the FA inhibitors. This is now measuring sphingomyelin and cholesterol in the type C mouse neurons using the same inhibitor. And again, one could see a reduction. This is in sphingomyelin, and this is in the cholesterol in these mice. There are more studies underway using multiple doses to see if we can improve these numbers. In this particular study, we just used one dose in these mice. So more studies are underway. This is now changing gears to the other receptor. This is CB2. So unlike direct CB1 agonists, CB2 agonists can be safely used without any psychotropic effects. And they've seen diverse effects, including protection from inflammation. JWH-133 is a prototypic CB2 agonist that's been used extensively in animal models of pain and neurobehavioral diseases. And it also crosses the blood-brain barrier. We've evaluated it in a mouse model of Farber disease, which is due to deficiency of acid ceramidase activity, and it accumulates ceramide. Farber disease is an excellent model of inflammation, and this is why we chose to use this agonist with the Farber disease mouse. This is showing CB2 in the cerebellum and cortex of 8.5-week-old Farber mice. On the top panel, this is the wild type. And if one looks at the lower panels, one could see the increased expression of CB2 in the Farber mouse. And this is in the cerebellum, these two in the cerebellum, and this is in the cortex. This is now looking at the liver, CB2 in the liver. Again, the top panel is the wild type, and this is the Farber mouse. And as I mentioned, these mice have lots of inflammation. So we're going to see very high expression of CB2 because these animals have macrophages in extensive amounts of macrophages in their tissues. This is now looking at CB2 of the same animals in the liver, and this is the top panel that were untreated, and one could appreciate the reduction in CB2 expression in the livers of the treated animals. We also looked at MCP1, which is the monocyte chemotactin protein, which is a biomarker for inflammation in these mice. Again, this is the top panel looking at the livers of the untreated. Again, one could appreciate the very high expression of MCP1, very high inflammation, and these are now the treated Farber mice. Again, this was only one dose, the same dose we had used in the Niemann-Pick studies, and we have more studies underway to evaluate now the lipids in these animals, the ceramide levels, sphingomyelin levels, in addition to see if we can improve the markers for inflammation in these animals. We also looked at the plasma in these treated animals, and one could appreciate, again, there was a significant reduction of MCP1 in the plasma of these treated Farber mice. Oops. Okay, now we're going to switch to a third disease, which is actually MPS, which is the disease that we study mostly in my lab. Looking now to see if there's a commonality with the other LSD diseases, Farber disease and Niemann-Pick disease. We wanted to see if the endocannabinoid system is actually affected in MPS. So first we looked at CB1 in the MPS IIIA mice, and we saw that there was a reduction of expression of CB1 receptors in the brain. This is looking at mRNA in 3 different sections of the brain. Remember, CB1 receptors are found on neurons. This is in the cerebellum, cortex, and the hippocampus. So we had the most reduction in the cortex of the MPS IIIA mice of CB1. We then looked at expression of CB1 in the same sections. This is the wild type animals of the same age, and this is the MPS IIIA mice. And one could appreciate that there is, again, a reduction of expression of CB1 in the same areas of the brain. Now we looked at CB2 in the MPS IIIA mice. This is looking in the liver. We looked at mRNA expression, and here we're going to have the opposite because remember, CB2 is a measure of inflammation. And we have an increase of CB2 in the liver of these mice. And again, this was confirmed by looking at expression of CB2 in the MPS IIIA mice. This is the wild type on top, and this is now showing the MPS IIIA mice. You can appreciate the extensive expression of CB2 in these livers. This is looking now at the cortex, wild type, and this is the CB2 in the same cortex sections of the MPS III mice. One could see that we have more expression compared to the wild type of the same age. We then looked at, we had some human 3A fibroblasts, and we treated them with two different CB2 agonists, and we looked at MCP1 in the media of these cells, and we found that there was a reduction in the MCP1 inflammation marker in the media of these fibroblasts. These studies are also underway. These are preliminary studies. We are now using, just like the other models, different doses, and we're actually going to be looking at different pathologies and doing different studies with neuronal cells. So I hope to have more results by our next meeting. So in summary, the ECS is dysregulated in several LSDs. We've now done some studies in type AB and type C Niemann-Pick disease, Farber disease, and MPS IIIA. And we found a commonality with reduced expression of CB1 and elevated expression of CB2. ECS acting drugs can impact pathology and clinical diseases in LSDs. FAAH inhibitors in type AB and type C mice have been used, reducing lipid storage and improving disease. CB2 agonists in Farber mice and MPS IIIA cells have now shown reduced inflammation. So our next steps is to continue to evaluate these drugs in other LSD models, including other models of MPS, and to identify some lead compounds for clinical trials in LSD patients. Thank you for your attention, and I'm ready for questions.
Thank you very much, Lila, for your excellent talk. And I think there are some questions in the chat, or one question. And someone is asking, do you think that this treatment with THC directly to the endocannabinoid system, could it be better than the enzyme therapy replacement in Niemann-Pick A and B?
Well, I think you mentioned treatment with THC. We're not going to be treating them with THC.
Sorry.
Yeah, we're going to be using the FAAH inhibitor because we don't want psychotropic effects. We don't think this is going to actually impact the storage in all the diseases. I mean, there was a reduction in the Niemann-Pick of sphingomyelin because we know that in Niemann-Pick disease, what actually happens is CB1 activation activates a neutral sphingomyelinase, which is what's causing the reduction of sphingomyelin. It's early to say anything about the other disease, but I think this could be an adjunct. I don't think this would be a sole treatment. I mean, maybe for Niemann-Pick it may, because it directly reduces sphingomyelin, which is what's being accumulated in that disease. But I think for diseases like NPS, I mean, we don't know if any effect on GAGs. We know it's going to reduce inflammation. So I think it would probably be an adjunct, not a sole treatment.
Thank you. Other question, is CB2 similar to CBD versus THC?
I'm sorry, I couldn't understand.
See, is CB2 similar to CBD versus THC?
Well, THC they're psychotropic.
Sorry, my mistake.
Yeah, Yes, the THC and CBDs could be psychotropic. I mean, what we're doing, these inhibitors are indirectly affecting these receptors because you don't want any psychotropic effects.
Thank you. And after your work, it's interesting to see that with this system we are acting not only in the nervous system but on the peripheral tissues. Who I think it could be applied in other lysosomal storage and lipid disease. There are
Definitely,
many diseases affecting the AAH. So thank you very much because it's a very, very nice work. And I think it's time is over. I think yes. And I would say that it was a very amazing and very exciting session. I think that's all. And thank you very much for all the speakers, and thank you all the people for your attention in the last day and in the last session for therapies. And I don't know if you want to say something, but I think time is finished.
Thank you.
Thank you.
Bye. Thank you.
Bye.
Thank you all the speakers for your work and your explanation. Bye.
Thank you.
Bye.